Okay, welcome to the Gut Club's second podcast, our second installment. And today we're going to be talking about a very deep subject. Everyone, we're going to be treading very deep water here. And it's all about bacteriophages. These are the healthy viruses that actually play a very important role in regulating our microbial balance. We're going to learn about this. And we're going to be learning about it with Linda Smith, who is a new PhD student at APC Microbiome, and also Adrianna Ponzanelli, who has her own group on Facebook. In fact, let me show you that. Let me first show you Linda's site. Oh my gosh, where are you? You know what? I'll show you that later. Linda, PhD student at APC Microbiome. That's a very impressive thing. We crossed paths on Instagram, and I'd like to say that I immediately recognized greatness when I saw your blog, eternalmicrobe.com. It's just fantastic. Everyone check out Eternal Microbe. APC Microbiome is a part of UCC Cork, University College Cork in Ireland. So we're very blessed to have Linda join us from Ireland today. Adrianna is with Ecotummy. And she's joining us from Southern California. So thank you both for being here today to talk about viruses and healthy viruses at that, and how we might be able to modulate them to improve our bacterial balance. That's what our focus is in the Microbiome Club. Actually, our group page, the Gut Club Stool Test Discussion Group on Facebook, is mainly all about bacterial balance. But today we're going to be learning about how viruses regulate bacterial balance. It's amazing. So we're happy to have Linda here today from Cork. Let me just say one thing about Cork before I give Linda the floor, and then Adrianna will help us to interpret what Linda is talking about. First of all, I recognized probably a decade ago that the work coming out of UCC in Ireland is leading the world, especially in the field of gut, microbiota gut-brain axis. We have amazing people working in Ireland, people like John Cryan and Ted Dinan. I'm not sure I'm pronouncing his name properly. I believe I am. People in putting out so many papers. They're so prolific when it comes to microbiota gut-brain axis. The most prolific I've seen on Earth. So Linda happens to be studying in a place that is quite enviable. Linda, what got you involved in microbiome science, and why did you choose bacteriophage as your,
I actually, thank you very much, Pete. Just want to say thank you very much. It's great to be part of your Gut Club. I actually have a slide on my journey to studying the microbiome. So yeah, thank you very much, Adrianna, also for tuning in. It's very early over there in California. Thank you so much for your time. Okay, so yeah, I thought I would give a bit of my background. So you're you're wondering, how did I come to be studying gut phagiomics? And you mentioned the gut-brain axis there. So my background was actually in neuroscience, but I was thinking today that we didn't have a module on the gut-brain axis, or the role of microbes in mental health, or the gut metabolomics back then. So it seems like a long time ago since I did that, but it's kind of crazy to think that there was no crossover with microbiology really then at all. But it goes to show you how new the field of microbiology is. And then afterwards I worked in a hospital lab for a bit, and then I changed gear and got a degree in computer science, and then I worked in industry for a wee bit, but this was in data unrelated to microbiology or bioinformatics or anything like that. And I was always kind of thinking in the back of my mind about genomics, and how can I look at cool bioinformatics data and study genomics. And then I had what I would describe as a microbial awakening in December 2019. So Keith has probably had this a long time ago, and Adriana, you're laughing, so you've probably had this light bulb moment as well. But mine was quite recent, and it corresponded with me getting a microbiome test kit, and I got it for Christmas, and then I sent it away, and I was just blown away by, you know, the composition of my microbiome, and like researching the different strains and what they do and their functions, and looking
Amazing.
at scientific, it's incredible stuff.
Linda, which test kit did you use, out of curiosity?
The Atlas Biomed one.
Okay.
Yes. Yeah. It was good. I have a blog post about that. They don't actually ship to Ireland anymore. I kind of grilled them on some questions, which made me kind of like, I kind of thought maybe I poked a hole in their pipeline, which I don't know where down. Then they're not. Anyway. But yeah. So this really kind of opened Pandora's box for me, and it's once you kind of discover the world of microbiology, it's really hard to, you know, focus on anything else. And March 2020 was quite disruptive from my timeline. I'm sure Keith is aware what happened then, but I moved back home and I wasn't really happy with my job. And I was just thinking, how can I like get from mortgage data to microbiome data? I was applying for lots of jobs. I happened to apply for a job in APC microbiome in a gut phageomics lab. And to be honest, at the time, I didn't even really know what gut phageomics was, but I was really lucky that Colin Hill's team took me on as a research assistant, and I was able to cut my teeth on some microbiome data. So you're talking about prolific researchers in APC, and I really do like believe in this team, and they're absolutely amazing. My mentor at the time was this guy, Andrei Shkoparov, and he is, I'm actually working under him now in my PhD, looking at bacteriophage. So yeah, don't want to bore you too much with details of my past.
Well, that's fantastic. I'm happy to hear that you had an epiphany.
Incredible.
Yeah. It's amazing. It's I'm sold on microbes now. I'm absolutely hooked. There's no going back.
Yes,
okay,
I bore all my friends and family the same way. Yes.
Me too.
Yeah, in fact, my son used to say, "Dad, I'm not listening." Okay. Okay.
Not another conversation about microbes, Mom.
Yes, that's right. That's right. Okay, Linda, please continue.
Yeah. Okay, so we know that bacteria run the world. They play really important roles in their environment, in the ecology of the earth, converting elements into biologically accessible forms. They're also super critical for our health, breaking down fiber and releasing nutrients, making vitamins available. Keith knows a lot more about this than I do, but hand in hand with bacteria are bacteriophage. So bacteriophage have been there evolving alongside bacteria this whole time, and phages are the natural predator of bacteria. So they're involved in this ancient evolutionary arms race, and where, you know, bacteria are evolving mechanisms to evade bacteriophage takeover and stuff like this. And one result that we gleaned from this is CRISPR-Cas9. So I'm sure you both have heard of that before, but that's an example of a bacteria defense mechanism against phage. So that just even understanding this biology and stuff that's going on has driven so much advances in biotechnology, and has just even like our understanding of genomics and biology, like at a genetics level, has been from understanding phages. So yeah, they're a really simple organism, but we still are learning so much about them, and there's so much more to learn. And they drive the evolution of bacteriophage, and they can change the bacteria physiology. So instead of kind of thinking about them as this parasitic entity that's just killing bacteria, if you really zoom out to the macro level, it's more about a co-evolutionary dynamic.
Yeah, one thing I just wanted to break in, I had a revelation myself just today, wondering about the relationship between bacteriophage and mitochondria, which were actually once considered to be bacteria and evolved to become part of the mammalian cell. Have you seen any research about the interaction between bacteriophage and our mitochondria?
Wow, that's really out there. I haven't thought about that before. I do know that there is research being done to see if like bacteriophage bind with eukaryotic cells and stuff. But I don't actually know too much about it. Could you tell us a bit more about that?
No, I don't think anyone does, so that's why I'm bringing it up. There's not much out there. There is evidence that the DNA, the mitochondrial DNA, are actually from phages. Anyway, let's crazy stuff. Continue, please.
Okay, so another thing that's absolutely crazy about phages that blew my mind is that they're the most abundant biological entity in the biosphere, and as well as that, they're the most diverse. But just to think that if you go out and you sample soil, or the leaf of a tree, or the seed, you're gonna find phages in there, and there's so many of them. So what are they doing in their environment? Okay, and they're obligate parasites. That's kind of the same as all viruses. They are dependent on the host for replication. And it's said that there exists at least one type of phage for every strain of bacteria. So that's really good for the phage therapy crowd, I suppose, and the therapeutic potential of using phages to target bacteria.
You know what? Let me break in there for a moment. Did you see the recent news? It was challenging the theory of the narrow host range of phages. I just saw this published January 17th.
Okay.
Yeah, there may be more than one bacterial host per phage.
Yeah, well, I could see that happening, to be honest. Actually, what I found interesting is that bacteria change their surface receptors. So they have like a genetic toolkit where they can swap out their cell surface receptors to evade the phages in their environment. So yeah, it's likely that I suppose even it's even more mental to think maybe the bacteria wants a certain type of phage to infect it, so then they express that cell receptor. I don't know. That's really out there.
It is I've been wondering if, for instance, Lactococcus bacteriophage can also infect lactobacillus, for instance. I think the research was focusing on staph bacteria, and phage being able to infect more than one type of staphylococcus. I need to get more into this. This was only published last week. So
really
anyway,
cool.
Yeah, pretty wild. Please continue.
Yeah. Okay, so so the main real biological feature of phage would be their life cycle, and this kind of lends itself to their to them being agents of gene transfer and drivers of evolution. So they have the typical lytic or virulent stage that we associate with most viruses, where they invade the host or bacterial cell, they hijack the cell's molecular machinery, and basically become a factory for manufacturing bits and pieces of phage, which are then assembled inside the host cell, and then they burst from the bacterial cell and go on to repeat the cycle again. And And then you have the lysogenic stage where the phage can actually integrate their DNA into the host cell's DNA and remain dormant until they sense a more favorable condition. There's different theories about, why they choose one state over the other. Obviously, it's just all brand new research. So yeah, take-home message, I suppose, would be that phages are incredibly ancient, they're numerous, diverse, and they're uncharacterized on a global scale.
Mhm.
Yeah, by the way, if you go back a moment, you talk about horizontal gene transfer. Is that playing a role in, what's really on the minds of a lot of people these days? That's antibiotic resistance in bacteria, and how phages may be driving that by, through horizontal gene transfer. What's your view on that so far?
Yeah, absolutely. I haven't read an awful lot about it, to be honest. I read a paper which I wrote on the blog a while ago. I'm just trying to remember the details, but I'm pretty sure they ran simulations, having lots of different setups, like, having phage with a bacteria that have an antibiotic resistance gene and ones that don't. And the outcome was and then they added they added antibiotics, I think, into the mix, but the only outcome where they both survived was when there was lysogenic phage. So the lysogenic phage were transferring the antimicrobial resistance, the antibiotic resistance gene to all the bacterial population.
Wow.
So it's kind of, yeah. So yeah, they're preserving themselves and their host. I find that amazing.
Amazing.
Yeah, there's a term I learned just, in preparation for this discussion. I think it's known as PAS, phage antibiotic synergy. And I think we're probably at the very beginning of just considering how antibiotics, which are so prevalent in the environment, are directly affecting bacteriophage. We can talk. Let's talk about that a little bit later in the context of birth. And the differences in both phage and bacterial populations in cesarean section versus natural childbirth. It's interesting to consider how phages may be responsible for those shifts based on exposure to antibiotics. Okay, I'm done now.
Great.
Okay.
Yeah, this is great. I love talking about this stuff. I love it. It's great. That's a new term. I've never heard of phage antibiotic synergy, so thanks for that. I'll have to check that out.
I was happy to discover it.
Okay, so how old, this is all just kind of like giving some relevance before we go into viral dark matter, because I'm coming from the perspective of phages. There's loads of other viruses out here, but this is the only really lens that I know. So I get very philosophical, or, I try to think when I think about viruses and evolution, I think about the origin of life and, try to think billions of years back and just about the origin of life and where it all started. But anyway, this guy, I'm not the only weirdo out there thinking about this stuff. This guy, Microbe, he's a French evolutionary bioinformatician, and he reconstructed this beautiful phylogenetic tree. So what he did was he just mapped the eggs to all these phylum of bacteria. He mapped the viruses that invade them, and he was able to trace it the whole way back. He also made a phylogenetic tree for archaea as well. So he was able to trace it all back, and he reckons that this evolutionary tree of life has viruses right back to the last universal common ancestor. So it seems that, you know, viruses have been around for billions of years, and this doesn't really tell us much because we don't know anything about the LUCA, but we only can know that the virus has been around for a really, really long time, and that we can presume that they've been even further back. So he says that the virome was dominated by double-stranded DNA viruses, and that because of the diversity that existed in one of these groups, it indicated that this realm of viruses had already reached considerable diversity prior to the split of archaea and bacteria. Okay, the phylogenetic reconstruction indicates that the LUCA was similar to the existing prokaryotes with respect to the repertoire of viruses it hosted. DNA viruses have evolved and diversified extensively already in the pre-LUCA era. And then this is some points that weren't in the paper, but I was kind of looking up this morning just for reference. So, well, we know that viruses predate humans by billions of years. We have around 8% of our genome is from old viral DNA. And also, I just read this kind of Pop Science article this morning. I just glanced at it, but we have some of this fossil viral DNA can become activated, and some viruses are capable of chromosomal integration. So viruses can insert their DNA into our DNA. So I just think that's pretty wild.
It is pretty wild. And can I make a comment about that? That actually fascinates me because a long time ago, I wrote a post about how viruses integrate into the genome and how we can find a spermatozoid virome. So when you, not even as a human, but also as a cellular level, you know, when the sperm is donated, it already has a virome. And if you want to read a little bit about it, sometimes I forget a lot of stuff. So I just go to viral dark matter and you type sperm virome or sperm and viruses, and then you can find the posts.
Yeah. Linda, may I ask a stupid question?
There's no stupid questions here. Keith.
Oh, thank you. Thank you so much. Yes, there are. How can we easily tell the difference, you know, between a bacteriophage and what's considered maybe a regular virus?
That's a good question. Well, I'd say phages have these hallmark genes, so they have a really distinctive morphology with their capsid and their tail. And I haven't done a lot of research on other different types of viruses, but I don't think they have these hallmark genes. So you can kind of like cluster viral sequences and say, okay, this one has a capsid protein, and you say, all right, it's a phage.
Okay.
Yeah.
You know, and by the way, you know, you talk about viruses entering the host genome. That's a very controversial subject these days regarding you know, SARS-CoV-2. And there's a paper that came out several months ago saying that it was possible for SARS-2 to enter the genome. I don't think there's any evidence of that happening.
Wow.
But it also makes people wonder about whether or not the spike protein induced by the mRNA vaccines is entering the genome. So, you know, this is very controversial material, and I don't think there's any evidence against it at this point. So.
Yeah. Well, yeah, I've kind of taken my foot off the throttle with the SARS-CoV-2 research, but I mean, I think it's possible. And this, I understand it's controversial from that regard, but like bacteriophage integrating their DNA into host bacteria, like that's fact, you know, there's no disputing that. So,
okay.
And I think also viruses, I mean, that's the whole purpose is that they are robbers of DNA. That's their whole purpose of reproducing. I would say living, even though it's controversial to say if they are alive or they are not alive, just because of the requirements to say the word, the semantics, living. But their whole purpose is to invade and steal. So they are just a big thieves. So I don't see why it's not a possibility, because I would say like any virus could integrate their DNA into any cell.
What's interesting, is the mechanism. It's called reverse transcriptase, and it got me wondering what enzymes would be needed to make that happen, and then I began considering viral bacterial interaction, and actually there's evidence that the enzymes could come from bacteria to allow the virus to enter the genome. But you also talk about how we can activate viruses and activate phage. Maybe later we can talk about what's called prophage induction and how we can use dietary strategies.
Absolutely. Yeah.
To induce phages and even inhibit phages. All right, your turn, Linda. Thank you.
All right, thanks, guys. Okay, so what is viral dark matter then? So I'm sure both of you are aware of our advances in sequencing technology. We have these amazing high throughput sequencing machines that allow us to, we can just take the sample from an environment, then we can see all the DNA from that sample or from that environment, and we can characterize it so we can understand what's there, the whole community's DNA. And viral dark matter essentially is these sequences that are of viral origin which do not align to any reference virus sequences. And apparently 40 to 90% of sequences don't have a match, of viral sequences don't have any match in a reference database. So that's quite interesting that we have no reference, we have no point of reference for these viral sequences. They're just something completely new that we haven't seen before. And I'd say there's like a whole revolution happening in this where people are discovering so many viruses through metagenomics. So we used to be really dependent on culture-based methods. And I read this amazing word in a paper the other day. It says bacteria can be recalcitrant. So recalcitrant actually means having an obstinately uncooperative attitude towards authority or discipline. So I thought that was just a brilliant word that I learned. So bacteria are really difficult to culture. And to culture the virus, you need to know and culture the host. This is with phages anyway. And so yeah, I have another slide on this later on, but metagenomics is only really, it's like brand new. So I found a fact online that it's been around since the early 2000s, but looking at viruses and virus-like particles this is only really a decade old field. And these protocols, like computational protocols for looking at viruses and metagenomic data, has only been around for the past 10 years or so.
I should point out that you're coming at this material, Linda, not just, as a research scientist focusing on microbes, but you're also a computer scientist and with the ability to write your own software. Isn't that right?
Yeah, I try.
Yeah,
I'm too clumsy to work in a lab, so.
That is so cool.
Yeah, it sure is. I don't want that to be lost on our audience.
Keith, can I just kind of translate a little bit what Linda said to our audience?
Please, and also at the same time, let us know what inspired you to call the group on Facebook Viral Dark Matter Study Group.
Oh, yes. So, well, just going back to what Linda said, just for, the sake of, everyone to understanding this content, what Linda is saying is that she is in the lab and now in the science of discovering unknown viruses that they don't infect humans or animals as per se, but they only infect the bacteria, which are other little, microbes. Viruses are one type of small little teeny microbes, and then bacteria are a totally different entity. So we didn't know a lot about what we know now until computers arrived. And it is the reason of computers that we are all of a sudden discovering a lot of things about, why kids with, for example, autism, might have some symptoms, because we couldn't really understand. We didn't know that a lot of these little teeny microbes existed because we couldn't really see them because, what we used to use to try to figure out these teeny little microbes were just lenses. We would look under a lens and then we would decide, oh yeah, there is a microbe or there is not a microbe, but we couldn't really understand a lot about them. And it was because of computers that we were able to discover, oh wow, we actually didn't know this existed. Why? Because some of them, they cannot reproduce in a laboratory with food. They might be just die with oxygen, or like Linda is saying, because these little viruses, they only can reproduce inside a bacteria, then we need to first grow the bacteria so we can actually grow the little viruses. And I hope I explained it well.
Yeah, that's really, really cool. In fact, Linda has a blog all about how crAssphage, which is the most abundant virus in our gut, was discovered using software. And I think that relates to, I mean, she goes into this in great detail. And I was really fascinated to focus on crAssphage. And isn't, Linda, is that the focus of your PhD work? It's taxonomy for crass-like phages?
It is, yeah.
And these are the phages that infect, particularly, apparently, the Bacteroides phylum. And that's really, I mean, it couldn't be more important. I think that you've chosen this area, this focus on crassphage. I remember when I first learned about crassphage, I noticed there was a capital A in the word, accentuating the ass part. And I wondered, what the scientists were thinking. Often they try to inject humor here. And I looked back in my my history of posts on Facebook. Even it was 2014, the year before I started the Gut Club, actually, and I posted about the crassphage on a page that focuses on humor, actually, because I thought it was crazy, but what and funny. Why did they name it crassphage? These viruses that are targeting, in particular, Bacteroides, and this is really important because it's reduced Bacteroides that is seen to be a hallmark in cesarean section and autism, and the focus is antibiotics as the cause of these reduced Bacteroides. So I'd like to, to monitor the future research on how these crass-like phages are affecting, both a rise and a fall of Bacteroides. Why did they call it crassphage with a capital A?
I actually they called it after the algorithm they used, cross assembly, not because of where it comes from, but yeah, I do think, why did they call it that? But it's hilarious. And yeah, you say that it's interesting that I've chosen this line of research. I honestly feel like it has chosen me in a way or something. Like I just kind of fell into it, and now I don't know, like I'm hooked. So it all just kind of happened. Wasn't like I was researching about the microbiome, but I didn't really know about the significance of viruses. And I suppose now when I tell people about what I'm doing, they kind of have this like visceral gut reaction. They just don't want to hear anything about viruses, which I understand.
Well, the fact that you've chosen crAss-like phages of everything, is especially poignant in today's world, because I like to focus from a phyla standpoint about microbial balance in bacteria and how that differs among populations across the globe, and how it might be a matter of environmental toxicity for raised Bacteroides, which can skew immune response. But we need to factor more and more, how this environmental toxicity is also affecting our crAssphages, and how that's directly regulating populations of Bacteroides. So
Yeah,
hats off to you.
That was something I really didn't understand, Linda and Keith, and I hope that you might help me to understand. So I was doing some reading and in crAssphages, and just to come and kind of like, know that what you were guys going to talk about. But so about the contamination of the lakes or the water, yeah, it seemed like when they find crAssphages in water, that's a sign of human fecal matter contamination. And I found that really kind of interesting to call it contamination or like of water, because on the other side, I thought, well, having these phages, it's a good thing, because if you don't have the phages, then you don't have the Bacteroides intestinalis. Then you cannot digest, specifically fibers that are sugars that we eat in our grains, because that's how Bacteroides intestinalis are more prevalent into Western societies, I think. So I was kind of confused, and I didn't know, well, is it a good thing, or is it just says that there is human poop in this pools of water, or is it a sign of infection?
I think they're just using it as a signature of fecal contamination. It depends where they're sampling it, though, I suppose.
Because crAssphage is the most abundant phage in the gut by far, is that right?
Yeah, absolutely. So I can head on into this now.
Okay.
So it is the most abundant one. It was only first isolated in 2018 by Andre and his wife Katia, who work in APC, and I just think these images are beautiful, these like 3D cryo-EM images, and there's going to be more and more coming out of those. I can't wait for it. But yes, it's the most abundant in virus in our gut, which is just mad to think we didn't know in 2014. I think this paper came out in 2014. So this guy Dutil, he, instead of just assembling reads from one metagenome, he pooled them all. So there was 4 pairs of female monozygotic twins and their mothers, and they pooled all the reads together, and then he found that like 50% of the reads in the human gut samples correlated really strongly with this crAssphage. They kept finding this long contig, so this 97 to 100,000 base pair like segment of our genome, and then I don't know how they resolved. They found like a DNA polymerase, or they annotated the genome, and then he blasted this crAssphage genome against like publicly available database records, and found that it was just so abundant. Like, it's such an amazing discovery, really. And this is a real triumph of metagenomics. So yeah, it's extremely interesting.
And it's not so surprising when we consider that Bacteroides at the genus level is often dominant. So I mean, there's so many types of Bacteroides, so it would stand to reason that there should be so many types of crAssphage. But isn't it something like 80% of all the phages in the gut are crAssphages? Is that right?
So I think it's about, well, actually, I have a slide a bit further on here.
Mhm.
Yeah, so it's 86.7% are crAss-like. Well, around. This was from Natalia Uten's paper in 2021. But you can read, this is like from mapping reads to the human gut virome. So there are vast majority of them are crAS, and then you have just very small 10%, 9.4% other viruses, Tevin, Viridae, 3.9%. So I don't know what they are, eukaryotic viruses or something. So it's really abundant.
Well, this is just an amazing window to helping people that are out of balance, whether they need to raise protective Bacteroides or reduce Bacteroides and raise protective Firmicutes. But through this window of crAssphage is a fascinating possibility.
Well, yeah, it's definitely, Keith. I think we need to understand more what they're doing, how diverse are they, try just to understand more so we can understand the relationship they have. There's so many experiments going on at the moment, where they are adding, like crAssphage or phage to like a sterile gut, or like the word escapes me now, but it's where you have mice that have been raised in isolation. Oh, they're germ-free mice. So they have like a few strains of bacteria in their microbiome, and then they add crAssphage in, or they add bacteriophage in. But you would think that the phage would kill all the bacteria, but usually what happens is the bacteriophage, their quantity increase, which shows they're replicating, and then as the viruses are having a spike or a burst, the bacteria are decreasing, but then they stabilize, and actually the bacteria levels go higher than the virus levels. So there's an agreement happening. It's there's some kind of symbiosis, or there's some kind of agreement.
Yeah, I've read that the infant microbiome assembly begins with higher levels of phages, and then bacteria even the score, so to speak. So
wow,
fascinating to consider that FMT, actually fecal microbiota transplant success, has been shown to be partially due to the phage populations, which is why I happen to have a hypothesis about breastfed baby poop being the best untapped medicine on earth, perhaps partially due to high levels of bacteriophage.
Yeah, that is true, because they have done these studies where they I think they heat killed the bacteria, and they only leave the phages. I think it's kind of like a sterilized FMT, and they have been able to recover certain diseases with this type of fecal transplant, which is not like fully diverse. It's only bacteria mostly killed. One question I have about crAssphages is to be able to have a phage in large amounts, you have to have bacteria in large amounts. So I want to ask Keith, what do you know about Bacteroides to be in the amounts? I know you always talk about the importance of lacking Bacteroides, and in relation to the 80% amount of what we're seeing crAssphages in humans, because I feel like the reason that we have the specific bacteria for the crassphages is because we have largely consumed mostly like our cereal consumption, from when we started farming has increased, differently to how we used to eat before agriculture. And I would love to see a difference in, more remote populations, African, very remote, populations, versus more westernized populations that we are eating, like, cereals and breads and all these fibers. So I was kind of interested in that, like what's the true reality in the 80% count of crassphages in different diets? Did I make sense or no?
Yeah, hasn't there been? I think there's been some research about low carbohydrate diets and how that, or not low carb, but different types of diets and how it relates to crAssphage.
Yeah.
Gluten-free diets, I think, has been studied. Go ahead, Linda. You answer that better than I can.
Yeah, sorry, I don't know any about that. Yeah, so, well, this was like a huge global study they did, but to answer directly about the diet categories, there was this, I think it's called a deep line research, where I think it was from Holland, where they had like 1,200 or so microbiome or metagenomes, and they analyzed, they looked for associations there, but there was very weak associations with several diet categories. And I looked up the supplementary data today, and it was like a very, very weak correlation with the your protein intake, but like extremely weak. You wouldn't write home on it about it, and there was no significant association with health or disease. So we think it's a benign entity for humans. So that's very interesting. I don't know. Maybe we need to. The thing about crAssphage is, and the virome studies in general, is that the virome is so individual specific, like it's so unique to the individual. I don't actually have a slide on that today, but I know Andre has done a study where they traced the microbiome or the virome of a bunch of subjects for like a year, 2 years, 3 years, I can't remember, but the all of their viromes are extremely individual, and they're all really stable as well. So over like a long period of time, you have your set virome. So I think, so in terms of raising your Bacteroides level or influencing your Bacteroides level through, some dietary intervention, which will influence your crAssphage levels, like we're well off that, because we need to understand individual people's viromes better. I think that's just my take on it anyway, but I do feel that we will, we have like this wealth of data, so we just need to, look more and analyze and ask these questions. And that's the beauty of bioinformatics and, sequencing. Like, it's incredible that we are able to ask these questions. We just need the time and the computer power.
Fascinating.
But yeah. So this was a big study. They looked at remote human populations. So they looked in Malawi, which I looked up today, somewhere in Africa, and they looked in the Amazonas of Venezuela, and those populations were crass positive. So they were trying to see, I think Bacteroides is associated with a Western kind of diet, I'm not sure. They wanted to see if it was like, a cosmopolitan, or it was just a Western type virus. But no, they found that it was in these remote populations. They also looked in these mummified gut samples of 3 pre-Columbian Andean mummies. and a European Iceman,
Wow.
but it wasn't positive. So they didn't find it in them. But that could be for other reasons, maybe the crass DNA wasn't stable over time, or it broke down, or there's stuff, I don't know. But it's interesting. They did also look in primates and old world and new world monkeys. So old world ones are from Africa and Asia, and new world are from South America. And they found that sequences similar to the ones in humans were in crAssphage ones were in these primates, which the authors of this paper then suggested that this virus has been evolving with primates and humans for millions of years. They also found that it's in crassphages in infants. It was previously thought that it was acquired over time, but yeah, they found it there. And then what I mentioned before about it being a benign entity, that there was no real strong association with diet categories or health or disease, which is very interesting. This also, it's very, crAssphage is very place specific. So this is kind of a confusing enough graph, but the size of the pie chart corresponds to the number of samples they got from the area. And so here's one in the middle of Australia, and what they did, I think, was they pooled the sequences, and then they compared those ones to all the sequences, all the other crAssphage sequences around the world, and they found that here that the sequences from this region are most similar to the ones from there, and there's rather than the small segment that is from the crAssphage sequences are similar to somewhere else in the world. Whereas over here, I think in San Diego, a bigger chunk of the pie of the crAssphage sequences, they're more similar to crAssphage sequences elsewhere in other geographical locations around the world. I mean, I chalk that down to maybe people traveling, or maybe traveling. It's probably a very busy place. Yeah. Or
California is so diverse. I would say I have never been in a more diverse place.
Yeah. Oh, I can't wait to go over there. It would be great.
No, you have to.
Hey,
Yes.
Linda, just to break in for a moment about infants. I did see some 2020 research showing that breastfeeding modulated the bacteriophage populations. And it's interesting to consider, these human milk oligosaccharides in breast milk are prebiotic to Bacteroides. So this would then modulate bacteriophage populations in infants.
Okay. I wasn't aware of that research. That's interesting.
Yeah. Okay.
So you were asking about the abundance. So in 2013, the most abundant virus in the human virome was unknown. 90% of all reads in the virus-like particle-derived metagenomes can correspond to crAssphage. 22% of the reads in the total metagenome, and 1.68% of all the human fecal metagenomic reads in public databases align to this crass genome, which is, and you were talking about Bacteroides a lot. I'm only here mentioning for the first time, I think, that the Bacteroides is the host of the crAssphage, and Bacteroides dominate the human gut virome. But also what's super interesting is that most of the bacteria in Bacteroides phylum remain uncultivated as well. So not only do we not know a lot about the viruses, about the crAssphage and the viruses, we haven't isolated a lot of the, apparently 99% of bacteria from this phylum they refuse to grow. They're recalcitrant, they're stubborn.
Yeah, I think this is a really controversial point that's being made here. In my research, I've found that Firmicutes dominate at the phylum level, but Bacteroides can dominate in a healthy gut at the genus level. So, you know, I like to think that we can, know what constitutes a healthy balance of microbes.
Yeah.
But I think everyone's still learning this.
Yeah, definitely. Yeah, go ahead, Adriana.
And one question too. So the crAssphage, is it specific to Bacteroides intestinalis, or as far as we know?
Well, no, there's lots of different species. That's the thing, is we don't actually know, and that's going to be, I suppose, part of my PhD is I need to collect, loads of metagenomic data, and we try to find out what is exactly a crAssphage. See, this is another kind of crux to the problem at the moment, is that we have very kind of, well, I don't know, apparently we have kind of like loose terms for what defines a crAssphage. It's like, okay, well, if 30% of its genome has these genes, then it's a crAssphage, but the taxonomy of it is still pretty murky. So because because bacteriophage they swap out their genes, and I have a slide on this in a bit, but so if you think about viruses, so think about the crAssphage order. So they're not just like a family of viruses, they're going to be this whole new order of viruses. So yeah, I've lost my train of thought there, but yeah. So
join the club.
Yeah. Oh wait. Yeah. Okay. So if you think about all the kind of like all the order of crAssphage, so you have this thing called a pangenome, and that's all the different genes and genetic modules that a virus can have access to. And because of homologous recombination and different methods of recombination, they can literally just like swap out these genes. Like, okay, so with humans, we get the genes, we got the homologous recombination during sex with like the gamete and the spermicide or whatever. That's it, like that's our germline genes. But bacteria and viruses are more fluid, like they can swap and change their genes through so many different mechanisms, and that's what makes microbiology so fascinating. It's so interesting to me, because it's so like when you look at the human genome, you're like, oh well, it's too easy, you know what I mean? It's like static.
Mm-hmm. Yeah, that really fascinates me, because it's a constant evolution that will never stop. We saw it in COVID, right? Like the viruses are constantly evolving to the point where, well, hopefully at one point it's going to become so weak, that it's not going to be such a massive problem anymore. But something that I really kind of get fascinated by is like how microbes that didn't exist, maybe I don't know if we didn't know they existed, or they actually never existed before until we found about them. Like there was a fungus discovered on the top of the Chernobyl reactor that nothing can live in there. Supposedly nothing is going to be able to live under those conditions of radiation, but somehow they discovered this fungus that evolved to produce so much melanin that was able to survive those conditions. And the same happened in the Gulf, spill, back in few years ago, where they discovered that there was a new bacteria. They don't know if there was new or already existed and evolved, but a bacteria that helped to eat or clean the oil from the sea. And I feel that this is like you say, we don't, it's we can go as fast, but I think they will go faster than us. So this is when I think viral dark matter is fascinating. When are we going to catch up? We'll probably never will be able to catch up.
Adrianna, viral bacterial interaction is barely, if at all, on the map in the public mind. You know, it's such an amazing thing to consider that with the pandemic, we're really receiving a lesson in viral bacterial interaction if we'd only pay attention, because it's these microbial microbes that are regulating the immune response. So, you know what, we've been going at this for a while. How many slides do you have left, Linda? You might have a ton, and maybe we should take care of a couple important ones and then wrap it up, and then do another, you know, have another presentation uninterrupted by you.
Yeah, of course, of course. So what would you what would you like me to do? I can briefly run through the horizontal gene transfer stuff and just the significance of trying to Adrianna, what you're saying there's about the extremophiles and the fungi is a whole other world. It's so fascinating. So yeah, I'll just, do you want me to just race through them now?
Maybe so, because I think we'll be here all day with all the interruptions. So
it's so interesting. Okay, well, these are just new papers that have come out. You know, that we're discovering thousands of new viruses all the time from the metagenomic data, and it's brand new. Oops, I'm just going the wrong way. Okay, I think we've already talked about this. 99% of the prokaryotic diversity in the biosphere from 16S RNA data fails to grow in the lab cultures. This is just a meme I found online, which is hilarious because bacteria in nature are so strong and defying the physical limits of life, like living inside the Chernobyl reactor and stuff, whereas in the lab, they are much more finicky and more challenging. So why are viruses so challenging to study? Viruses have no universal marker gene, whereas bacteria have this really highly conserved 16S or RNA subunit. All bacteria have this, and yeah, it's easy. It's highly conserved, so you can tell by, you know, a small difference in one bacteria or another what kind of bacteria it is. Okay, so the life cycle of the bacteriophage, the prophage is where it's the integrated DNA into the host cell. Phage encoded recombination. So this is a mechanism whereby phage can recombine their DNA. So homologous recombination is the exchange of nucleotides between two DNA molecules. So when I kind of think of a simplified infection is where there's just like one phage and one bacterial cell, but it's not like that. There's probably millions of bacterial cells and viruses, and so this can happen where two phage inject their DNA, and their DNA can recombine, and then you get these recombinant phage genome, and then you get like this hybrid phage that's a combination of different genes.
That is so cool.
Yeah. Also, there's this is from a genetic and engineering paper, but and this is like just how they are making these recombinant phage genomes in the lab for genetic engineering. But I kind of was thinking that maybe also recombination could occur between. So if you imagine that this segment is the bacterial chromosomal DNA, and then this green bit is like the prophage. So maybe an alien or an invading phage DNA can do recombinant recombination with this prophage. I don't know. That's just me. That was just a thought that I had. I don't know if that's actually been confirmed. So this is kind of like just like a little visual of how the virus genomes are mosaic, and that like structurally they can be so different because they can swap genes in and out. So so crAssphage, like if you have a crAssphage that are the same species, or similarly related ones from the same family, actually, they would probably have like a very, very similar genome composition, whereas distantly related phages could have very small amount of similarity. And these genes are modules that are exchangeable among the population. And this is not unique system to viruses. It's also prevalent in bacteria. And then you could have like the genome where there's a huge number of genes whose function is not known. So this is kind of more like in reality what it's like in the crAssphage genome. So you have, so you can scale up those little genetic segments into to these genomes with way more segments, which you can group according to replication, assembly, or transcription. There are different functions, and there's a lot of genes that we just don't know what they do. I suppose I can skip this one for in the interest of time.
Linda, just sorry to interrupt you. This is just fascinating. Just one question. You said that so to distinguish what's in the sample, you have a 16S, and then you classify them as bacteria, and then however you classify fungi. And how do you know that these are bacteriophages then, and maybe not something else that you're discovering?
Yeah, so that is something that I need to learn, but we have programs like VirSorter, so you might be able to get like, so say you have this whole phage genome, okay, and you do an alignment to reference databases of known viruses. So we might get a hit for this little C segment, and they might say, okay, this segment is similar to like a tail protein or a capsid protein in our database. But that doesn't really tell us about like the whole, that just because this, 3% of the genome is similar to a phage tail in a database doesn't necessarily mean that makes this whole phage, like a lactococcus phage or something. So we would still have all of these other genes which are uncharacterized. That is my understanding of it. It could be wrong. And yeah, so we do, a lot of this has to be proved experimentally as well. So we have software and stuff to predict the genes and the proteins. But we need, like I say, it needs to be predicted, or needs to be verified in the lab. Okay, so what's the point of all of this? It kind of advancing our understanding of phage biology, what is selecting for or driving these instances, what is causing all these interesting phenomenons of the phage genome, and then evolution and taxonomy, and predicting genes and proteins. Of course, we can mine, because of all this viral dark matter is new, we can mine this genetic material for brand new genes and proteins, which we don't even know what they would do yet. So to wrap up, so phage in the future, there's probably going to be phage-based therapies, for medicine and agriculture, for drug resistance, long-term therapies. Phage are going to be used as vehicles for drug delivery, clinical, okay, well, once we get like the green light for clinical trials and the safety and efficacy of bacteriophages, like I say, phage therapy isn't really my wheelhouse. I'm more into the ecology of it and trying to just understand what it is they're doing in the first place. But hopefully in the future, we will uncover a beneficial role of phage in the gut. And then we can have a more holistic view of the microbiome, and we can understand how we can, safely treat conditions where we're not disturbing the natural ecology and harmony between phage and bacteria. One can dream anyway. That we can,
Linda, I was researching several months ago the idea that we can apply probiotics directly to the lungs, in context of of COVID-19 and discovered there's quite a lot of research in nebulizing probiotics. And then I stumbled on a paper about nebulizing bacteriophage for delivery to the lungs. That could be a future.
That's cool.
Future.
I take these ones. I take this Floraphage, and they are a combination.
Oh, wow.
Yeah, they're a combination, and I sell them in my store. I take they're like a combination of like your ordinary phages, and I think what they do is that they kill E. coli and maybe other pathogenic bacteria, and they cross-feed good bacteria. So good bacteria would eat the proteins, or
Adrianna, all the bacteriophage products I've seen on the market, everything on the shelf is all targeting E. coli. But there are places out of the country where you can order more customized phage therapy.
Yeah, they have 4 different types. It's like Cyphobiridiae, Miroviridiae. There are other 2. They're like 4 different ones.
Yeah, I think it's all called Prefer Pro is, I think, the main cocktail being sold in a few different products. And I wonder if it's not counterproductive in some areas, since E. coli is actually a good thing. You can.
The different types of E. coli, but it apparently targets pathogenic E. coli. And okay. I must say I have other different brands of phages, and I have tried a lot of phages from the market, commercial. I have skin phages, a bunch of phages, but these ones have worked for me and my clients. Wow. I don't know what is different about Ardo. He's a medical physician, but he makes excellent products. And, this is the one that actually has helped me. If I take 2, I just feel it usually, but what they say is that it just potentiates the benefit of your probiotics. And I don't know what's the difference, but,
All right, I'm going to share a screen. Go ahead,
I'll have
Linda.
to interview about it, Adrianna, sometime. It sounds very interesting.
Sure, that would be lovely.
All right, I want to share a screen and wrap things up here. So here is Linda's beautiful blog. I love the name of it, Eternal Microbe. What inspired you, Linda, to name your blog Eternal Microbe?
It's a good, Keith, you're too kind. Thank you so much. This is just cool. This is just a hobby stuff that, it's on a dusty corner of the interweb where nobody really visits. So thank you so much. I'm glad you appreciate it.
Here's your book, yes.
Oh, yeah. Well, Linda
that's
wrote
amazing
a blog
book.
about it.
Yeah.
It's so good.
Yes. Yeah, that really opened my mind to, living in a microbiome or a macrobiome. It's amazing.
How did you have this, epiphany to name your site Eternal Microbe? Because it's a beautiful, catchy phrase.
Yeah. Well, I just thought that microbes will win. Microbes are always going to be here, no matter what happens to us. They're going to be around, and their lifespans. Also, like a singular bacterial cell never dies. Like I remember having a conversation with someone and it blew my mind. So if a bacterial cell has enough nutrients and stuff, it can just live indefinitely forever, whereas we get old and die, but microbes their longevity is unreal. They can get frozen in ice and stuff for millions of years and form spores and stuff, and they're just they're eternal.
Yeah. But we're all going to die. Thank you so much, Linda, for that. I'm just kidding. Okay, so here we have Echo Tummy. This is Adrianna's website, and she does gut health coaching, and here is Adrianna's Facebook page, Viral Dark Matter Study Group on viruses and phages. And let's see here. This is something that, an article that was from 2020 about how we can, use foods to landscape our microbiome by the way we trigger phage production. That's called prophage induction.
Oh, and Keith, we should talk about phages and viruses good viruses in your kefir, because there are some studies about viruses in kefir.
Interesting. I need to learn about that myself. Thank you, Adrianna. But this paper is something Linda and I have both, even before we crossed paths, we're looking at. And Adrianna, I posted this a couple of years ago on your Facebook group page, and I was so fascinated by this particular graphic, and and how we can use certain things to both inhibit and raise phage production. And here's one that I'm just looking at.
Prophage induction, yeah.
Yeah, I'm looking at this just more so today about how pomegranate and cinnamon relate to Bacteroides, and this is a really important protective Bacteroides species. So to think that cinnamon and pomegranate can basically slow down, it's a negative value, the phage induction that may preserve Bacteroides. So
yeah,
I think
absolutely.
that's so interesting. And then bee propolis, I'm a fan of, is actually raising the induction of phages related to Enterococcus, which is also associated with autoimmunity. So
that is amazing.
Yeah, it's fascinating stuff. A couple other things I wanted to share real fast. Here's the Gut Club's page about breastfed baby poop as a donor for FMT, and some discussion about the first thing I listed was healthy phages may be a key to success. And I wondered about how fresh stool may be a better donor material, because freezing can deactivate or injure phages. Linda, can you comment on freezing our bacteriophages?
I think they can be frozen, yeah, because when the stool samples are taken, they're frozen immediately for later analysis, I think.
Okay.
Yeah. So they're preserved, I think.
Okay. All right. Well, that's interesting. I'd like to see more about that. All right. So let's see here. I think that's about it. Here's the article on breastfeeding from 2020, modulating the virome and phages. But I think we should wrap this up. And I want to thank both of you today for this primer. Basically, I think we could have talked all day. I still feel like an infant myself, speaking of infants in this area. So it's great to have an expert in the room, a couple of them. And Linda, I wish you the best in your PhD. I'm going to eagerly monitor your progress.
Me too. I'm so excited to find more about crAssphage and all your work. And
thanks so much.
Yeah.
Yeah. So thanks to both of you.
We should definitely do this again. Yeah. Thank you very much.
Thank you very much. So Adrianna in California and Linda Smith in Ireland. You're in Southern Ireland, right? Are you in Cork now?
Yeah, I'm in Cork now.
Okay, fantastic. I remember 1983, I hitchhiked from London to Dublin and then down to Glendalough, but I didn't make it all the way down to where you are. What a fantastic place.
Maybe I'll catch up with you, because I am probably going, my husband is from Cork, and I just got my Irish paper. So I'm going to go visit you maybe one time.
Definitely, please do.
Yeah, my husband gave me papers, brought papers from your university. I don't think I could enter your university, because I wouldn't, I would need to study many years to get in there. But I was fascinated that they had a big microbiome art exhibition where there was all this, you know, paintings about guts and like microbes. And Shay brought me stuff from Cork, and he's constantly saying to me, we should go to live to Ireland and you can go study, or, you know.
Yeah.
I'm like, I'm too old.
Adrianna, I didn't know Shay was from Cork. I knew he was from
Yeah.
Ireland, but I didn't know it was Cork. That's serendipitous. Okay.
Yeah,
With that,
my whole family, my whole, you know, Irish family are living in Cork, and we actually don't have family in Pleasanton. We don't have anybody. And we've been talking about going back to Ireland.
Aw.
Definitely hit me up if you ever come over here.
That'll be lovely.
I
Hit
don't
you
know
up.
if you can. Yeah, I
they're
don't know if you can hitchhike from Florida to Cork. Maybe.
No.
That'll be great. Oh my God,
Yeah.
Keith.
They're Linda, they're not going to just hit you up. They're going to be sleeping on the floor in your living room.
Oh yeah, I'm sure I can accommodate you somewhere in my little
Okay.
hovel here.
That'll be so much fun. Keith, let's go.
Next time we'll learn more about Linda's explorations in the field of botany and talk about viruses in the soil, which is something I know nothing about.
That'll be so cool.
Wow.
Yep.
Okay. Thank you both. And
Thank you, Keith, for hosting.
And I hope everything stays well for
amazing.
everyone. Okay. Bye-bye.
Yep.
Stay in
Bye-bye.
touch. Bye.